Hi, and welcome to episode 69 of the Breaking Bio podcast. I'm Morgan Jackson, a PhD student at the University of Guelph in Ontario, Canada.
Hi, I'm Tom Housley. I'm a technician at the University of Cambridge in England.
I am Stephen Hamlin. I'm a postdoc at the University of Southern California in Los Angeles.
And today we're joined by Dr. Sandra Rehan, who's a new assistant professor at the University of New Hampshire. Thanks for joining us today, Sandra. You want to tell the listeners at home a little bit about yourself and your work?
Hi, yeah. I'm Sandra. I'm new faculty here at the University of New Hampshire in the Department of Biological Sciences. I've been here almost a year, and I work on bees. So pretty much all things bees fascinate me. I study things ranging from biodiversity and conservation to population genetics and phylogeny, and have delved into comparative genomics and a lot of behavioral genetics. So very diverse research foci, but everything centers around native bees.
So we're not talking honeybees. You're one of the few people who aren't working on honeybees, but rather on all the other bees that are generally more interesting. The way I see
Yeah,
this.
I'll study pretty much anything besides a honeybee. That's correct.
Awesome. So you do a lot of stuff, and we'll try and touch on it as much as we can throughout the course of this interview. But I know from growing up with you in the academic world that you do a lot of work on the evolution of sociality, especially within the carpenter bees. Correct? Mm-hmm.
Yeah, that's probably my biggest passion, is to study why did sociality evolve, how did it evolve, when did it evolve, where did it evolve, and I use the carpenter bee subfamily Xylocopinae as a great model. So this group evolved over 100 million years ago and is ranging from solitary to eusocial and back again. So it's a very interesting group to study historical biogeography as well as social transitions and character state evolution that way. And it's a great opportunity to travel because they're found all over the world. I've had a really great time doing a lot of work done in Australia, Borneo, Africa, Panama, all over. They're found everywhere, and they vary in their behavior markedly between each location and each species. So definitely an interesting group.
So I was looking at your website there, and I saw that you have been doing some work on aging and gene expression. I was just wondering, so I guess most people probably don't think of insects in terms of aging, let alone things like changing in gene expression profiles with age. So maybe can you just give us a little overview about the work you're doing there?
Yeah, so everything has a lifespan. Of course, an insect often is shorter than a human, but over their life they experience very different environments, and they do senesce just like everything else. And so we've been looking at the bees to see over their lifespan, how does gene expression vary, how does physiological processes vary. And so we've been able to do kind of genome-wide gene expression studies, largely looking at the brain, to see how these bees are changing and finding very interesting patterns, changes in innate immune gene regulation. So they shift in their immune function as well as different metabolic processes. So it seems that they rely on protein or carbohydrate stores at different points of their life. As well, the small carpenter bees often overwinter in very harsh kind of Canadian winters or New Hampshire winters. And so during these times, they really have extreme cold hardiness and freeze tolerance. And so we are interested in how they're able to do that. So, I mean, over their 1 year of their life cycle, they experience a lot more dramatic and plastic environments than arguably a lot of mammals will ever experience. It's a crazy world when you get down to the genes at that point. And we can it's really fun now because we can do genome-wide studies and really not have a candidate gene in mind, but rather ask, at any given point in the colony cycle, at any given life stage, what is going on within the insect.
That's really cool. So you said about the physiology, but do they change the sort of social behaviors as well?
Correct. So yeah, we can look at it on a gene basis, on a physiological basis, but also on a behavioral phenotype basis. So the bee I study the most is this small carpenter bee, Ceratina calcarata. It's found in North America on the East Coast. And it ranges from kind of a solitary stage early in the colony cycle, so females will forage and found nests on their own, and then, but they're very good mums. They stay on the nest, they guard and groom and inspect and care for their offspring through development, and so this is more accurately called subsocial. The subsocial just means prolonged parent-offspring interaction and parental care. And so, but then later again in the colony cycle, it gets a little weirder yet. They not only interact with their offspring, but they produce different kinds of offspring. So they produce regular daughters who overwinter to become foundresses or solitary females in the next spring, but they also produce dwarf eldest daughters who are small in body size, depleted in fat stores, first to eclose, and are also then forced out of the nest to forage and feed their siblings prior to overwintering. In doing so, they not only experience a very different social environment than the rest of their siblings, but they don't overwinter successfully because they're small, they're depleted, they're, for various reasons physiologically and behaviorally, they're not making it overwinter, so they're not actually surviving to have any reproduction of their own. So it's a true case of worker-like altruism in what we thought was a solitary bee. So it's a solitary bee turned subsocial turned wannabe eusocial. But yeah, so they're very complex behaviorally throughout this colony cycle as well. So mothers are experiencing very different environments, and the offspring receive different allocations of food and stimulus, and thereby they're also treated quite differently. So it's a very plastic group in terms of everyone involved.
So you were saying just before as well about the protein and carbohydrate kind of ratios being important at different life stages. So how does this affect, what they're doing each time, like with behavioral wise or reproduction?
Well, it's early days in terms of our understanding, but we think that carbohydrate metabolism is very important for a lot of the foraging they do, and they really rely on the kind of sugars in the nectar. As they're consuming it, they're really living off of kind of superfluous sugars. But often during overwintering and things, they're living off more of their fat stores and some even protein degradation. So they're actually relying on very different food stores, much like other organisms do. And so we see that they, in their short lifespan, are experiencing very different nutritional requirements and depletions.
So you're officially calling these subsocial, and yet it almost sounds like they're on the cusp of having almost like a caste system with your minor, slightly weaker siblings being reared. At what point does this go back to be considered, or maybe not go back to, but to be considered, truly eusocial or the next step up from subsocial?
It's a interesting paradigm because we have these gradations, and this bee doesn't fit in any of them very well. A lot of bees do throughout the colony cycle experience maybe a solitary and then a social phase. That's kind of common in bumblebees, for example. There'll be solitary foundresses making workers, and then they're eusocial. But here we have a bee that is definitely subsocial, we can argue, this prolonged parent-offspring interaction. They're not truly eusocial because to be truly eusocial, would require reproductive division of labor. So if there was a reproductive phase for which these dwarf eldest daughters were coexisting, then that would be eusocial. However, they're not really helping raise siblings, they're kind of helping overwinter siblings. So it's really a technical definition. It just doesn't really fit in any of the categories that well. I've argued they're at, the evolutionary is kind of like the brink of eusociality. All the behaviors are there, it's just the life history isn't quite matching up with what we commonly perceive as being a eusocial insect. So I think natural history and behavior is much more varied than some of the pigeonholes we try to fit the organisms into, unfortunately.
For sure. So since you're pretty well buried in the world of eusociality and the evolution of eusociality, there's been quite a bit of debate in the last few years over kin versus group selection, spurred by a couple of fairly high basis names. Do you have your own theory on what's going on? Are you in either base camp?
I don't think it's good to be in a camp. I'm a bit of a naturalist myself, so yeah, I think there is a lot of standing theories and a dearth of empirical evidence. I think I think there's strong evidence for all sides, and I don't think there is. Unfortunately, this unifying theory I don't think is what we've actually found, and so I think there's a lot of room for more natural history and more species and more realization that our descriptions and our pigeonholes are not necessarily describing all the complexity we see. So, for example, I study a species in Australia, another small carpenter bee, that's both solitary and social in the same populations at the same time. And so this is a very nice natural experiment. I can ask the cost and benefits of sociality, controlling for phylogeny and species and ecology and all of these things. So this is the same species in the exact same spot. And so I studied the species for about 6 years there and asked, what are the costs and benefits of sociality? So actually empirically assessing, kin selection or Hamilton's rule. And I found, in fact, that solitary bees on the whole have higher fitness than social bees. And so, arguably, then why is sociality being maintained in this population? One can posit that maybe under very harsh and extreme ecological conditions, this would select for a social grouping. And there are some advantages, some predator defense and some survivorship benefited by being in a group. But empirically, what I measured over the study didn't correspond to that. So it's hard sometimes. You have this disconnect between theoretically what might be and empirically what you observe. And so I think the lack of long-term studies and the lack of unique species like this has led to there being a question, how universal, how unifying are things, and really we just need more data, and then we can really assess and test. So in general, there's been a lot of support when studying really highly eusocial things, that kin selection does explain it because they tend to be really highly related and they tend to be really efficient because selection has driven them to be these super colonies and super efficient. However, some of these groups that I study who are on this brink of sociality, who are dabbling on both sides, sometimes it's less clear the costs and benefits and the advantages. So maybe there's not a kin selected benefit and maybe there are alternative explanations. So I'm really gathering a lot of interesting data that doesn't necessarily fit all the normal theories. So I study kind of a unique group, which I find fascinating, but also makes me question, what do we know? And if we study another species, what are we going to find? So it's an exciting time, I think, but unfortunately also highly controversial.
I think that's a pretty good way of putting it. Exciting yet incredibly controversial. Especially if you're the bigwigs who are throwing the heavy punches in the big journals.
Yes.
So some of you touched on it a little bit about how much traveling and how you're studying a lot of these things in the wild. I hadn't realized that you were doing that much traveling. I knew you had spent a lot of time in Australia during your graduate work, but I hadn't realized you were doing so much natural history research, in Africa and Borneo and all the other places that you rattled off there. Is this all observing these things in the wild and trying to understand what's going on with these systems?
Yeah, largely. This genus Ceratana, the small carpenter bee, all notes and natural history notes on them from back in the 60s and 70s said it's a solitary bee with unusual social tendencies, or it's an unusually solitary bee or something, and they would always describe it to being solitary. I know there's some weird peculiar nests where there's a couple females hanging out or whatever, and so I knew there was something more to the story. And so when I went to Australia originally, it was under the guise of a single note in '62. Charles Michener had discovered multi-female nesting in the species, but again, it was so few nests, and it was really just a note. And so I knew there was something to that, so I wanted to explore it further. And then diving into it, we've got hundreds, if not thousands, of colonies, and we can actually assess things in a statistical way. Likewise, traveling, I was fortunate to do some work in Borneo, under there just kind of a natural history collecting trip to do some phylogenetic expansions. But in doing so, I found a lot of nests, from 4 different species, and again finding, hey, they're social. These bees are not solitary. There are multiple females in a nest, cooperatively reproducing, and with, remarkably, division of labor. So interestingly, whenever I do find these social colonies, one female is reproductive and the other is not. One female is forager and the other is not. And interestingly, unlike the honeybee, where you have a reproductive queen and a foraging worker, here we have a strange division of labor. We have a reproductive forager, so a complete control freak, and then usually a lazy sister who does neither forages nor reproduces. So it's a division of labor, but kind of in a strange way. So there's cooperative breeding, and this kind of secondary female, in most cases, remaining in the nest as a guard. So it's providing anti-predator resistance and protection, but not actively foraging like most workers do. And so we found that in Australia, we found that in Borneo, I have evidence for that in Panama. So it's interestingly, throughout the tropics, they tend to be more social, and then kind of in the cooler climates, we tend to see more subsocial and into almost solitary behavior.
So are you able to quantify in any way the fitness or the reproductive success of the females that are employing these different roles and see how they differ across them?
Yeah, so we just have a paper, it'll be out next month, I guess, in November, in Animal Behavior, looking at this. So in Australia, this natural experiment I explained where we can look at costs and benefits. And so we've actually quantified that empirically and shown that a solitary female actually has higher fitness than a social secondary. So these females who are non-forager, non-reproductives, seemingly waiting at the nest for some kind of an inclusive fitness benefit, don't seem to be getting that benefit. So it's kind of curious why they're staying. So yeah, we've done work on that in nature, and I'd love to do more of it. Unfortunately, you need kind of very large data sets to ask. I think that's probably why there's not many of these such studies. But yeah, so we have this one coming out, and yeah, I would love to do more of the same.
So is that across lots of different species, or is that within one?
That's actually one species that's both solitary and social in the same population at the same time. It's an unusual circumstance, but it's actually the most appropriate species to ask that question. But yeah, one could also do this across species if you had more data, but we just don't at this time. Yeah.
Getting that would be quite an undertaking.
I'm not dead yet, so we'll see how we go.
Well, that brings us to an interesting point. So you're just starting out as a new professor. You just got your job, you said, in the last about a year ago at the University of New Hampshire. Seeing as I'm a grad student still, and Tom and Stephen, before he had to leave, are kind of in that spot between grad school and being the PI, what are some of the things that you are maybe finding now that you're sitting in the other side of the desk, perhaps?
Yeah, it's there's no actual way of knowing. There's no like job description per se. I mean, you come and it's an empty office and an empty lab, and you it's kind of fun. It's the ultimate shopping spree in terms of setting up everything. But I've realized you can't do anything until you have all of the things. So until you have everything you need, you really can't get anything going. So the first few months it was really just setting things up, and I mean I already had so many ideas and projects on the go, so it wasn't a black hole in terms of that regard. You kind of already know what you need to do your job. You've been doing it for a decade or so, you know. But it's just a matter of finding it and dealing with the reps and getting the stuff. And it's a bit time-consuming, it turns out. And so you do that, and that's kind of phase one. And then phase two is really grant writing, and so you do that a lot. And so it's different. I mean, it's certainly more administrative than I would have imagined. You know, you always hear about professors complaining about administration, but you don't know, really appreciate it because it's not in your realm, you know. But yeah, so there's just more of that than I would have thought, but I didn't really know what to think because you're kind of coming in naive. But yeah, largely it's just been a lot of fun, and so I've actually got help for the first time. It's really cool. I mean, I have ideas, and instead of me having to be like piddling away at the desk doing 1/8 of what I would love to do that summer, I could do it because I can get 8 people to do it, you know, and it's a really fun group. I have, my lab exploded very rapidly. UNH has a wonderful group of undergraduates. It's very much like University of Guelph. It's a land grant school, so you have a lot of emphasis on farming and sustainable agriculture. And so there's a wealth of undergrads just really looking to get engaged with research. And so I was, you know, they're all knocking on my door, hey, can we work on bees? And I'm like, yeah, sure. You know, so I couldn't accept everybody, but it was wonderful to have help, and they enjoy it. They're sponges. They want to learn. And so we've been doing bee biodiversity work. We've been doing habitat preference work, floral association work. Just so little is known about native bees in New Hampshire, but even really in North America. And then I've been fortunate to get some grad students in the lab, and they're helping out with a lot of the behavioral genomics work. And so we've just got, it's kind of a team. We can go out in the field together, and we can share the data, and it's nice, you know. So it's largely been a lot of fun. It's just amazing how much more you have to multitask in a day. There's not like the day where you just sit because you're by yourself and you can just kind of work through things. You probably have to be doing 5 or 6 things all the time, and so I'm cool with that. But yeah, it was just, yeah, so there's really no job description, so it's kind of a choose your own adventure, because you are the boss, which is nice, but also you're responsible for that, so it can be, you know, you don't want to mess that up. So largely I'm having fun, but also very much realizing the responsibility involved.
Yeah, that would be the biggest divergence, I would seem to think, is instead of just, you know, having to look out for yourself and make sure your stuff is being covered, is now you have actually people counting on you to keep keep the money flowing and keeping, you know, a lab over everybody's head. So that's definitely got to be one of the bigger, trickier things. Have you started teaching yet, or is that off your table for now?
I'm teaching right now, and teaching a grad seminar right now in ecology and evolution. So it's a good group. We have a very diverse faculty here, and so the grad students all have different points of view, and research projects are very varied. So ranging from marine biology to genomics to behavior to like ecology and invasion biology. So it's a very mixed bag. So it's really fun to get them all in the same room, because their take and spin on things are very different, and their backgrounds are very different. And so I've enjoyed it. I mean, it's only been a few weeks, but it's been a lot of fun.
So I just wanted to, on a slightly different note, ask you about, there are some pretty amazing photos of bees on your website, and Morgan and I are both quite keen macro photographers, so I was wondering how on earth you got those.
Some of the photos are not mine. I've tried to credit them when I could. Sam Drogi at the USGS, he's a famous bee photographer. I think featured in National Geographic, and he's also has a really great Flickr site. And he has done wonderful things with bee photography, and with the black backgrounds and everything. They're quite fantastic. So those photos are all his. And so, but yeah, I mean, I also am an amateur macro photographer as well. And so I mean, that's one thing I could buy, a really nice macro lens. And I felt that was important as a naturalist, that if we could take pictures of the bees on the flowers and we can communicate the beauty and diversity. I mean, it's not a hard sell. Bees are a charismatic fauna, so.
You're the boss now, so. You don't have to justify it to anyone.
No, and I think it's great. And so yeah, if we can just take a photo, just show off how cool they are, you know, it's a little world, but it's quite complex and diverse. So.
That's awesome. So that brings up one good point that maybe we'll touch on before we finish up here. Is your research focus spans the natural history fieldwork all the way through to the whiz-bang comparative genomics world, which are generally two ends of the spectrum that you don't see meeting in a single research lab or with a single researcher either. Do you think these things are going to influence or, you know, meld together, you know, in a single lab better than they would on their own?
I hope so.
Sorry, I guess that was kind of a judgmental thing on your.
Terrible mistake. Yeah, no. I mean, I was a naturalist first, and I always will be. I think if you don't understand the organism, you can't ask appropriate questions. So, I mean, you can break down the life cycle, and you can break down the behaviors, and you can look at the phenotypes in a much more intimate way and a much more informed way because you have seen it and experienced it. You know, we're still a bit naive because I'm a big human and they're a little bee, but, you know, I think I feel like I know my organism better than most, and I take a lot of pride and passion in that. I really want to know what they're doing in a day. I have to know what they're doing in a day, and so doing that, for me, that's the most important thing. And then in terms of methods to ask interesting questions, so we can do behavioral assays, and we can do gene expression assays, and we can do all of these things, but really, for me, it's always coming back to the behavior and the natural history and the life cycle and the evolution of the organism. So I definitely would call myself an organismal biologist. And yeah, you learn methods to make it work because there's an interesting question you want to know the answer to. So genomics is a really fun tool, and it's come down in cost quite a lot in recent years. So it actually is amenable for a lab to sequence a genome, and so we're doing that. We have a genome center here, and we can actually just take bees from the field and crush them up and say, hey, sequence this, and you get more data than you know what to do with, and you figure it out. And so I think it's important to add in the contemporary techniques. Obviously, you can't just do natural history and only rely on that. I think integrated biology is the way of the future. I mean, a lot of people approach that through massive collaborations, and I do collaborate with different scientists, including right now I'm working with Brendan Hunt at the University of Georgia, and he's helping me a lot learning about DNA methylation and things like that. And so I think it's a time to be collaborative, but also if you have a vested interest in a history with an organism, I think that's important. And I can't speak for most labs, but I think for me that's the strategy, and I think combining natural history with modern techniques, I think, is very powerful. And so I would hope more people are doing the same. But yeah, I think, you know, being too isolated and too one or the other, I think, may result in less insights instead of more. You know, you want to know more. You want to get deeper, not shallower with your system.
Cool. I think that's as good a place to end it as we can find. So can you let everybody at home who wants to follow along with your research and now booming career, where they can find you online and keep up with all the work that you're doing in your lab?
Yeah, so we have a lab website. It's unhblab.com. And so that, I mean, it's not a blog or anything. I just once in a while update that. And I'm on Twitter. It's just at Sandra Rehan. So I figure you guys put a link to that if you want. And yeah, my email is just my name, sandra.rehan@unh.edu. And I'm happy to talk bees with anyone, so that would be great. And so yeah, we're at the University of New Hampshire, and we're just getting going, and it's so far so good.
Awesome. Like Sandra said, we'll definitely be putting links to all of her research and personal lab spots up on our website, which you can find at breakingbio.com. And of course, you can interact with us online at Breaking Bio on Twitter, or you can find us on Facebook by searching for the Breaking Bio podcast. I'd like to thank Sandra for joining us this week and talking all about her work on bees, and we'll see you next time.